welcome the next speaker we have, who have earlier spoken on section's role in strengthening AOAC international activities. Yes, that was Dr. Kaushik Banerjee, and he is the current chairman of the India section of AOAC International, and being a member of the Food Safety Standards Authority of India's scientific panels and working groups on pesticide residues and method of sampling and analysis. He regularly contributes to the development and implementation of food safety standards in India. So with this, I would like to invite again on screen Dr. Kaushik Banerjee, co-chair AOAC Committee of Section. And this time he is coming up again to speak on the case study of melamine, cyanuric acid and DCD in milk and milk products. So can you please have the AVA?
Hello everyone. I hope you are thoroughly enjoying today's conference. In this presentation, I will be sharing a brief update on a recent endeavor that AOAC India section has taken up to develop and validate a method for simultaneous analysis of melamine, cyanuric acid and dicyandiamide in milk and milk products. A brief note about project background. You all are aware that melamine and cyanuric acid are low-mass nitrogen-rich compounds that have been linked to protein adulteration in various foodstuffs in the past. So always these came in the media and it created a havoc across the globe about the adulteration and the health hazard that melamine and cyanuric acid had created. While melamine and cyanuric acid are not individually toxic, what happens is that when they come together, they form an adduct, and this adduct is formed basically because of hydrogen bonding, and the product is melamine cyanurate. Melamine cyanurate produces sharp crystals and it gets deposited in certain internal organs, for example, mainly kidney, and it causes damage of the internal organ or failure of the organ, and that's the reason the concerned animal dies. A similar compound named dicyandiamide is also used to minimize the environmental impact of grazing livestock, and it was found in trace amounts in dairy products in New Zealand. So that is the reason why these three compounds, that is melamine, cyanuric acid and dicyandiamide, are thought always together, that whenever we would like to test it in milk and milk products, probably we need to test all three, and best possible way is to test them simultaneously so that a laboratory throughput can be maintained and the test is basically commercially viable. So here are certain regulatory limits. The maximum level for melamine in Europe is 2.5 milligram per kilogram, and the same level is also applicable in certain other countries. Since melamine, cyanuric acid, dicyandiamide, these are basically food contaminants. So that is the reason the term maximum residue limit is not applicable. It is always called as maximum level. In infant formula, the maximum level is 1 milligram per kilogram. In Canada, this level is set at 0.5 milligram per kilogram. Codex Alimentarius Commission, on the other hand, has set a lower limit, which is 0.15 milligram per kilogram in liquid infant formula milk, which is also implemented in some countries. So currently in India, we are following the Codex standard. These contaminants are regulated at this particular level, which is set by Codex. Now, if you look at the review of various analytical methods which targets estimation of melamine, cyanuric acid, and dicyandiamide, you will see that in most cases earlier, the methods targeted two compounds, that is melamine and cyanuric acid. And the reason was obvious, because when this issue was brought into the notice of global community, and then it was understood that these two compounds were the major problem creator, because they combine together, form the crystal, and that causes damage of kidney. But since the issue of dicyandiamide was also reported from certain places in milk and milk products, So then it was decided that in every case, the methods were subsequently modified to include dicyandiamide also in the analytical workflow. So in all cases, one problem is there that these are the small molecules. They face a lot of trouble in extraction, in chromatographic detention. So that is the reason in many cases people have reported poor recovery. So to rectify that problem and correct the recoveries, stable isotope analogs of these three compounds is important to utilize. And it's also important to see that whether chromatographic separation or a longer retention can resolve this. So lots of research and development activities are going on, but fortunately stable isotope analogs for these compounds are currently available for purchase. And when you use it, then it can rectify the issue of recovery. But on the other hand, these compounds are pretty expensive. So that is the reason activities are also going on to see whether it is possible to avoid use of such isotopically labeled expensive internal standards. So in our laboratory at National Research Centre for Grapes, we earlier found that certain plant growth hormones like chlormequat and certain quaternary ammonium compounds, which are small molecules, very polar, and we tried to avoid their isotopically labeled internal standard by using special chromatographic columns, LC columns, and optimizing the gradient program in such a way that the target compound has adequate retention and it elutes at a longer retention time. So in that case, in many cases, we found that we could avoid the high matrix effect, because of which people talks about usage of internal standards. So here certain update, if you look at review of literature, the choice of solvent for extraction varies widely. It's important to precipitate protein, so that is the reason acid is used. Shaking samples with solvent and subsequent centrifugation appears to be a popular practice. That is the general workflow of sample extraction in all cases. The other approaches also include microwave assisted extraction and accelerated solvent extraction, and these are reported by various authors in various laboratories across the globe. Regarding cleanup, you see that these compounds have ionic properties, so that is the reason in many cases the cleanup method has prescribed usage of ion exchange cartridges, especially in solid phase extraction mode. So in literature, we see usage of mixed mode cation exchange SP and anion exchange SP cartridges for melamine and cyanuric acid analysis. But here one problem is there, that if you do one process by cation exchanger and another process by anion exchanger, and you are basically splitting the extract. So in this case, you require to undertake separate analysis. So that proves to be a little inefficient and not so viable, because it takes a longer time. So people are trying that how best it is possible to analyze these compounds simultaneously. So in recent past, most methods that focused on milk and dairy products, they either dropped the SP cleanup through these ion exchange cartridges, or they tried dispersive solid phase extraction cleanup, which is a popular technique for most of the food contaminant analysis, because that is easy. You just take an aliquot of extract, add the dispersive solid phase extraction reagent, vortex, and then centrifuge, and then the extract, whatever supernatant you get, that appears to be much cleaner for analysis by LC-MS. LC separation, traditionally people tried various phases. For example, when people tried reverse phase chromatography, problem is that these compounds are not having adequate retention. Many cases they get eluted in the void volume itself. So that is a big problem for their analysis. So people tried various novel stationary phases, which has certain functionalities that allows a better retention of such compounds. And worldwide analysis of polar compounds, ionic compounds is challenging. So we love to work on that and basically trying best to improve the retention of the compounds by using various innovative stationary phases and by modifying the mobile phase programs. The other options, recent past, various mixed mode uh, LC columns have come up in the market, but this hydrophilic interaction liquid chromatography is appearing most important and most popular because they are specially designed for retention of such compounds and provide adequate retention, basically that resolves the high matrix effect issue. Once the compounds are LC separated, final measurement is always by tandem mass spectrometry, and you know that in food safety, these are very adequately defined that whenever you use LCM SMS, each and every compound should be monitored for two multiple reaction monitoring transitions, and ion ratio deviation has to be also monitored, and that should not uh, uh, exceed a 30% limit when you compare with the standard in matrix. So this is the brief outline of the method and also the target outcome of the project. So at first, we would like to establish the method for simultaneous determination of dicyandiamide, melamine, and cyanuric acid in milk and milk powder by SMS. Both the major matrices will be covered. The calibration curves are expected to have a linearity between 5 to 200 microgram per liter with a correlation coefficient not less than 0.995. These are the recoveries expected as per the standard for all food contaminants. We should expect a recovery of 70 to 120%, and relative standard deviation should be quite good so that we have adequate precision in analysis. The limit of quantification as per the current regulatory requirements is expected to be minimum 0.02 milligram per kilogram for milk samples and 0.05 milligram per kilogram for milk powder samples. The method should be simple, rapid, sensitive, and suitable for qualitative and quantitative analysis of dicyandiamide, melamine, and cyanuric acid, and in a wide range of milk and dairy product samples. Because unless the method is simple, rapid, and uh, probable for regulatory compliance, these will not be accepted. And a routine testing laboratory expects that the method should not be complicated. The proposed LOQs are based on the present regulatory requirement. I've already mentioned about that, but there is definite scope to improve the LOQs so that in future, if there is a change in the regulatory limit, the method should also be applicable. Here, some preliminary data I am showing that we used the UPLC BEH amide column, 1.7 micron particle size, and this is the gradient program. Uh, uh, we used triple quad LCMS instrument, and here you can see that each and every compound has been monitored for two transitions. In certain cases, we can even use three transitions. So basically, these are providing more and more confirmation and high sensitivity analysis. So these optimizations are currently over. We are getting pretty good signals, and we are getting good sensitivity as per the limit of detection. And here I will show this after this slide. So this is the brief sample preparation workflow to start with or begin with. This we have taken from this particular reference. So what is done here, at first the sample is weighed. Approximately 1 gram of sample is taken in 50 milliliter of polypropylene tube. Internal standards are added here. Then water with formic acid. 10 ml water containing 0.1% formic acid is added. Then it is shaken, then homogenized appropriately. Then it's centrifuged. From after centrifugation, an aliquot is taken This aliquot is diluted with 9 ml of acetonitrile and again shaken. Then from this you take the aliquot. This is subjected to cleanup using primary secondary amine and C18 solvents by dispersive solid phase extraction cleanup. So you have to basically take the extract, put it into some tubes and add these weight quantity of the dispersive solid phase extraction solvents. So after vortexing centrifugation, the aliquot appears to be quite clear. That this clear supernatant is drawn and injected into LCM-SMS for analysis. And here I have already mentioned that a BH amide column provided a very good method performance, but we are still trying various other options. So here you can see the chromatographic performance. The first signal is for dicyanidiamide. It is followed by melamine and then cyanuric acid. So this is the elution pattern. And so this chromatography clearly says that all three compounds could be estimated simultaneously. We have set currently a runtime of 10 minutes. So in this 10-minute program, each and every compound are separated chromatographically and they are appearing at uh, in sequence of I think dicyanidiamide, then melamine, and then cyanuric acid, and the last signal is melamine. So uh, these are the chromatograms in solvent. DCD signal comes at 1.22 minute. This is followed by cyanuric acid, 2.38 minute, and then melamine at 4.52, 4.62 minute. And here you can see the chromatograms in milk matrix. So we could achieve recovery more than 80%. When you use a solvent standard, because of high matrix effect, the recovery is not consistent. In certain compounds, recoveries are a bit poor. But what we found that although recovery was poor, it was consistent. But here, when we used the matrix mass standard, the recovery could be corrected to a great extent, but still we are facing certain problems of inconsistency. So we are also going to use internal standard in this and trying to optimize the method in such a way so that it is acceptable and implementable across various laboratories of regulatory agencies. So these are the next steps. We will further optimize the method, as I mentioned, by using internal standards, by optimizing the chromatography mode, using different columns, different chromatographic separation possibilities. We will follow the Sante guideline for further experiment regarding analytical method performance. So as per the guideline, the final method will be evaluated for its precision, sensitivity, linearity, accuracy, matrix effect, and all other aspects. The accuracy of the method will be studied at minimum 3 level. LOQ is the compulsory requirement. Then at least 2 higher levels, we should check the recovery and the precision. Minimum 6 replicate study will be conducted. And we are expecting to complete the SLB study in the next couple of months. So I think it's a steady progress, and we hope that India section will be successful in completing the study well within the stipulated time period. So this is a brief outline of today's talk. This is one of the activities which India section is taking up. In addition to that, India section is also doing one study on multi-element analysis. This study SLB is already completed. This SLB study has already been communicated by the authors to Journal of AOAC International. It is currently under review. And India section has initiated the multi-lab study on that multi-element method. So that will also come up very soon, and then these will be implemented, and these will be prescribed to FSSAI for implementation through the regulatory process and inclusion in the FSSAI consent manual. So thank you very much. Appreciate your attention.